Hey everyone, welcome back. In our last video, we solved the jigsaw puzzle. We used BWA to align our tiny sequencing reads against the reference genome, and we generated a file called a SAM file, right? But we left off with a cliffhanger. A SAM file is a plain text. That means humans can read it, but it takes up a ridiculous amount of space. If you align a whole human genome, that SAM file can easily blow past like 300 gigabytes or something, right? So even if you are lucky enough to be doing this on a high-end workstation with like a huge amount of RAM and massive storage, but trying to analyze a raw SAM file will still bring your computer to its knees. So today we fix that. We are going to introduce the most ubiquitous tool in all of genomics, which is called Samtools. It is basically the Swiss Army knife for alignment files. We will use it to compress, sort, and index our data so it runs at lightning speed. Okay, so let's jump into VS Code terminal. Let's us first like start WSL. Okay, so SAMtools is a command line utility. So we install it just like our other tools. Okay, so we will run sudo apt get install SAMtools. Okay, enter the password and okay, press yes, and it will install SAMtools for you. Okay, once installed, type SAMtools and hit enter, and you will see a massive list of commands pop up, right? You can use it to merge files, pull out or like specific chromosomes and calculate statistics. But today we only need three specific commands, which is view, sort, and index. Okay. So first of all, our first goal is basically compression. So we need to convert our bloated text-based SAM the sequence alignment map, okay, into a compressed binary BAM, okay, which is binary alignment map. Okay, so computers read binary instantly. To do this to do this conversion, we will use samtools view. Okay, so what we will do is we will write this command samtools uh, view capital S small b name of the sam file, which is align results dot sam, and the name of the converted file unsorted results dot bam. This S, capital S means the input is SAM, and small b means output should be BAM. Okay, so hit enter, and this will take a few seconds. And on a real data set, we are using a very small data set here, for example. So what you get is an unsorted results dot BAM file, which you can find over here. Okay, so if you try to open it, this BAM file in a text editor, okay, you will see, or if you use the head command on it, your screen will just print out absolute gibberish, as you can see here. Okay, it is no longer human readable, but your computer loves it. Okay, and the file size like shrank about 75% or something, I believe. If we can see here, uh, I don't know if I can see it here. Okay, never mind. So the file size reduced a lot actually. Okay, you can check it out by performing it yourself. So, so we have a BAM file, right? Are we done? Basically, no, because right now the DNA reads inside that file are completely random. Okay, so when BWA aligned the data, it just wrote down the answers as fast as it could. Read 1 might belong to chromosome 10, read 2 might belong to chromosome 1, and read 3 might belong to chromosome X. It is completely out of order. So before downstream software can find mutations, the data must be sorted strictly from left to right across the genome. So we use samtools sort command. Okay, so let's run that. Samtools sort, and the name of the unsorted file dot bam. Okay, and output will be sorted results dot bam. Okay, this that O flag specifies our final output name. This step requires some serious computational lifting, so it might take a few minutes on a real data set. Okay, so let's run it. But as we have a very small data set, so it basically finished in seconds. But if you had a real data set, it might take a significant amount of time. When it finishes, your sorted results.bam is beautifully organized from the first place of chromosome 1 all the way to the end of genome. Okay, you still cannot see it because it's a binary alignment file, but your computer will love that file basically. Okay, so one pro tip is that once you have your sorted BAM, you can delete the original SAM and the unsorted BAM to save your hard drive space. Okay, if you will not be needing that file in the future, obviously. Okay, so we are now at the final step. Do you remember when we ran BWA, the very first thing we did was index the reference genome so the computer could search it quickly. Okay, so we have to do the exact same thing for our sorted BAM file. Okay, so what we will run here is Samtools uh, index, then name of the sorted file, sorted results dot bam. Okay, this command runs very quietly, but if you type ls to list your files, you will notice a brand new file called sorted results dot bam dot bai. Okay, let me see, it's over here. Okay, this file, .ba file PAI file is the BAM index. It is a tiny mathematical map of your alignment. Also in format you cannot read. Okay, so whenever you load your BAM file into a viewer or a variant caller, it will secretly look for that .bai file to instantly jump to specific genes without reading the whole file. And there you have it, the golden trio of Samtools: view, sort, and index. Okay, this converts your raw alignment into highly optimized production ready format. This sorted BAM file is the ultimate prize of the sequencing pipeline. With this file, we can finally do two amazing things. First, we can look at the genome visually, and second, we can run algorithms to find the exact mutations that cause diseases. So we will be doing those fun parts in the next video. And yeah, if this video helped you conquer the command line, please hit the like button and subscribe to the channel. Let me know in the comments if your SAM files have ever eaten up all your hard drive space. And I'll see you in the next video.